Thank you. I'm Hyeongseop Kim from EN001. Today I'm going to talk about EN001, which is an allogeneic mesenchymal stromal cell therapy, including its concepts and our recent clinical trial results. So here is my disclosure. And recently, the focus of drug development has shifted from the small molecules and antibodies to the cell and gene therapy. These advanced therapy medicinal products, or ATMPs, are now recognized as next generation modalities with potential to treat incurable diseases. The first ATMP was approved in 2003, and since then, especially after 2017, the number of approvals of the CGT products were accelerated. Although this figure does not include every approved ATMPs, but it clearly shows that how the development of this field has been accelerated in recent years. In 1991, Professor Arnold Kaplan first introduced the term MSCs to describe multipotent stem cells isolated from adipose tissue, bone marrow, and umbilical cord, etc. Later in 1991, Pittenger and colleagues reported that MSCs commonly differentiate into adipocytes, chondrocytes, and osteocytes. In 2005, the International Society for Cellular Therapy proposed the minimal criteria for defining MSCs, which are still widely used today. The MSCs are known to originate from the perivascular cell. When tissue injury occurs, they become activated and modulate inflammation and promote tissue regeneration. On the right, you can see Drs. Kaplan, Prokop, and LeBlanc, the pioneers who defined the characteristics of MSCs and explored their clinical applications. Although MSCs were initially considered stem cells, it is now well recognized that their main therapeutic effect is mediated through tropic activity. MSCs have stem cell properties, but their clinical effects mainly comes from paracrine signaling rather than direct differentiation. This has led to debate over whether they should be called mesenchymal stem cells or mesenchymal stromal cells. In 2019, the ISCT concluded that both terms can be used, but the choice should reflect the purpose of the study. As explained, mesenchymal stromal cells can be isolated from various tissues and exert therapeutic effects through paracrine actions. One unique feature is that MSCs do not express immune phenotype molecules on their surface, which allows their use as allogeneic cell therapy. Over the years, the numerous studies have demonstrated that allogeneic MSC therapy does not trigger immune rejection in the host. To date, a total of 16 MSC-based therapies have been approved worldwide, and 10 of them are allogeneic products. The field of MSC therapy has been largely driven by developments in Asia, especially Korea and Japan, while Europe has only one approved product authorized in Germany in 2021. Since the first clinical trial using MSCs were reported in 1994, more than 1,000 clinical trials have been conducted worldwide. Across all these trials, including those published in leading journals such as JAMA, The Lancet, and Nature, no tumor formation has ever been observed, and no serious adverse events or deaths have been reported. This extensive clinical history strongly supports the safety of MSC-based therapies. Based on this background, we developed EN001, an allogeneic Wharton's jelly-derived mesenchymal stem cell to target CMT1A and 1E. The Wharton jelly is a part of the umbilical cord. In CMT1A mouse model, EN001 improved peripheral nerve conduction. You can see the upper panel. Both motor neuron and sensory neuron, the conduction velocity and action potential was increased at week 4 and week 8. And also you can see the road behavior test at week 5. The EN001 increased the latency to fall up to the 40 seconds at the blue line. And also we captured the CT scan and also electron microscopic analysis. So the myelin sheet of the CMT mouse is thin, but it became thicker after EN001 treatment. This is the video of the road test. On the far left, you can see the untreated CMT mouse struggle to stay balanced, falling off after only about 8 seconds. Next, the mouse treated with 5,000 cells of EN001 shows improvement about 51 seconds. And next, the mouse treated with 50,000 cells, it maintains balance for around 96 seconds. So as you can see, EN001 significantly improves motor coordination in a dose-dependent manner. So far, we conducted 3 clinical trials in Korea. The first one is phase 1A trial for CMT1A. Total 9 patients will receive a single intravenous injection of EN001, 3 for the low dose and 6 for the high dose. The principal investigator is Dr. Byung-ok Choi in Samsung Medical Center. This is the primary outcome and secondary outcome measures. The major objectives are to evaluate the safety and tolerability of EN001. As exploratory efficacy endpoint, we used CMTNS, FDS, ONLS, and 10-meter walking test, etc. This is the CMTNS version 2, was developed by Dr. Shai and his team. It includes the symptoms, signs, and neurophysiological data from CMT patients. Lower CMTNS scores means that indicate better outcomes. This is the key inclusion and exclusion criteria. Although the patients who are diagnosed with CMT1A, the disease severity should be mild to moderate. This is the timeline. MSC was administered intravenously. The toxicity evaluation was at week 4, and safety and efficacy assessment was for 16 weeks, long-term follow-up up to 48 weeks. This is the result. There's no dose-limiting toxicity, infusion-related responses, and serious adverse effect. Only 4 mild adverse events were reported, and those are not related to the EN001. The right panel is exploratory efficacy data. You can see the only high-dose patients show a decrease from baseline to week 48. In some cases, the disease severity shifted from moderate to mild. After completing our phase 1 trial, we conducted an in vivo study evaluating the repeated injections. These results showed increased myelin thickness, reduced ratio, downregulation of PMP22 expression, increased myelin protein 0 expression, and improved gait and balance, long-lasting functional recovery in gait and nerve conduction. This is the phase 1B trial for CMT1A patient, the repeated injection. Is still ongoing. EN001 was administered 2 times at 4-week intervals. The total of 6 patients participated. Of course, there's no serious adverse event, but only 1 patient reported moderate and mild adverse events, but of course, they're not related to the EN001. Surprisingly, even if at low doses, the CMT1A score dramatically decreased and remained for 24 weeks. The result of the high-dose cohort will be available in the fourth quarter of this year. This is the summary of two CMT1A trials, and no dose-limiting toxicity and serious adverse events among 15 patients, only mild to moderate and transient adverse events reported, but no MSC-related. Therefore, we could say the single and repeated administration of MSCs were safe. In efficacy side, the high-dose single administration of MSC showed significant improvement, and repeated administration also showed better therapeutic efficacy. And the last one is the investigator-initiated trial for CMT1E. It's a single injection. Also, the principal investigator is the Byung-Ok Choi in Samsung Medical Center. Only 3 patients participated. Okay, yeah. These are the, okay, let's go to the result. Also, this is safe, and you can see the CMT1A version 2 score. Even if there's only 3 patients, but they showed a dramatic decrease of the score at week 8 and week 16. It's very happy to share an important update of Korea's regulatory pathway for advanced regenerative medicine. Korean government made a program that allows the use of regenerative medicine based on prior clinical research to treat the patient. This system aims to provide new therapeutic opportunities for patients. So we think we will apply for this program, and if approved, the treatment could begin early next year. This is the take-home message. Mesenchymal stromal cell therapy has a long history and have been proved to be safe. And therapeutic mechanism of MSC is trophic mediator. At the current stage, only EN001 has secured efficacy data from patients with CMT type 1A, 1E. And EN001 will be the first treatment available for CMT1A patients worldwide. Yeah, thank you.
Thank you very much. Yeah, we just have time for one short question.
How long do the stem cells stay alive in the body?
Yeah, that's actually in human there's no data, but in preclinical study we measured only 4 weeks survive in the body. But the MSC is the main therapeutic mechanism is paracrine action. So most of cell we inject intravenously, most of cell trapped in the lung, but they secrete the exosomes or protein and microRNA. Those are circulate whole out of the body, and it showed a therapeutic efficacy. So in clinical trial, we observed along 1 year or more than effects.